Mohrenweiser Lecture. This lecture was established through a generous gift by Harvey and Elizabeth Mohrenweiser to address moral and ethical points between the practice of faith, science, and religion. And by the way, I'm Ray Pickett. I'm the rector here at Pacific Lutheran Theological Seminary. So today we are very pleased to have Dr. Arvin Gouw give the lecture on the CRISPR revolution and the various uses of CRISPR, which is a powerful gene editing tool. I was just talking to Dr. Gouw about this. I didn't know this, but he was actually the TA to Dr. Jennifer Dauda here at UC Berkeley, who is her pioneering research in RNA is what set this whole revolution in motion. So this was when he was an undergrad, right? Is that right? Or graduate student? Yeah, master's student. So, and then Dr. Ted Peters is here, who Arvin is also known since he was an undergraduate. So Dr. Dauda, some of you know, won the Nobel Prize for that research. And so that's for this gene editing technology, which has tremendous potential for good in terms of gene therapy and other uses in healthcare, but also raises significant ethical and religious questions regarding the use of genome editing for reproductive purposes and the limits of human intervention. We were just talking about recently, I just saw an article this week of a doctor whose name I've forgotten, Dr. Gouw will know, who was just released from prison for actually doing that. So it's a real kind of interesting challenge. Dr. Gouw is currently working in the field of theology and science at the University of Cambridge, Faculty of Divinity, and I think is uniquely qualified to speak with us about the moral and religious significance and implications of CRISPR, as well as the science. He's also a scientist who until recently was at Stanford University School of Medicine. And he has also done research fellowships in science and religion at Harvard Divinity School and Princeton Theological Seminary. And I just learned this morning that he's living in Virginia now, but he's actually doing a second PhD in theology there. So again, uniquely qualified. Dr. Gouw received his doctorate from Johns Hopkins University School of Medicine, his master's degree in philosophy from the University of Pennsylvania, in theology, degree in theology from SMU Ecumenical Institute of Theology, in endocrinology and neuroscience from UC Berkeley. So a very diverse background. And so we're also pleased to have this morning Dr. Ted Peters, who's Emeritus Professor of Systematic Theology here at Pacific Lutheran Theological Seminary. So we welcome him, and he will give a response to this. They are working together. They edited recently a book called Religious Transhumanism and Its Critics. And then they have a book they are finishing together. Dr. Peters and Dr. Gouw are finishing an edited book to be published this summer called The CRISPR Revolution in Science, Religion, and Ethics. So please join me in welcoming Dr. Gouw, who will give the lecture. And then after the lecture, Dr. Peters will give a response, and we'll have some time for Q&A. So welcome, Dr. Gouw. Yeah.
Thank you so much, Dr. Ray Pickett. And first, I'd like to thank the Mohrenweiser family for this opportunity, for this invitation. I'm very, very glad to be here. Of course, whenever we get an invitation, I would check, what is this lectureship all about? When I saw that it's about, the moral implications of inventions in science and medicine and how it relates to faith, I thought, this is awesome. Every seminary or every university should have this kind of lectureship. Then I thought, but who would deliver the talk, right? So I'm very gracious for the opportunity, but this is one of those things where, as a scientist, I'm trained to focus on just one gene amongst 20, 30,000 genes. And here I'm forced to talk about general ideas and how it relates to religion and ethics. This is not my comfort zone. This is a gracious invitation, but I'm here to tackle the challenge and sin boldly. So that'll be the task today. And I'm glad that it's also at PLTS. This is home for me, Berkeley. I grew up in the Bay Area. I went to undergrad here. So PLTS has always been a campus that's very engaged with science, thinking of the luminaries in the Lutheran tradition, even from Pannenberg, Phil Hefner, Ted, it's very, very sensitive to the need of the public in engaging scientific advances. So I'm very happy to be back here. All right, so this is the title of the talk. Again, it's very ambitious. I think most of it will be science. I decided most of it to be science because I know the audience will be more qualified to talk about the theology and ethics than I am. It's always a nice trick. Let's see. Oh, that way. I'm trying to change the slides. All right. So the talk will be in 4 parts. CRISPR mechanism, maybe only 2 slides or so. Some applications, a lot of applications, the dangers and regulations, because I usually get asked about how is this thing regulated. So CRISPR initially was a, oh, did I do something? Okay. CRISPR was initially, oh, the pointer doesn't work. It's not on TV. So the CRISPR usually is, it was discovered as a bacterial immune system, right? And what that means is if we get sick by COVID, you know, we have white blood cells to fight off the pathogens, right? The virus, the bacteria. But if you're a bacteria and you get attacked by a virus, what can you do? You're by yourself as a single cell organism, right? You don't have white blood cells. You are a cell, right? So this was the research. This was Jennifer's research. And she found that, well, you have this kind of CRISPR-Cas system. Basically, if a foreign DNA, viral DNA comes in, it gets chopped off, and the bacteria knows it. The next time it comes around, it'll send that RNA along with a Cas9, a scissor, to basically degrade the virus, the DNA. Okay, so that's how it works. And they discovered this a while back, a decade or so before Jennifer realized that, well, hey, you know, this sequence, maybe I should take this. Is this okay? All right. So, so wait, the sequence, what if we put our own guide RNA? Then it's not just going to target viral genome, but it can target any gene we want in the DNA. That's a simple idea. Well, it looks simple, but they got a Nobel Prize for it, right? So, so this is them on October 2020, Nobel Prize, and this is the crystal structure that he just published 2 years ago in Science, of course. The blue part is the DNA, the white part is the Cas enzyme, the scissor that clips things off, and the red part is the deaminase that can do the nucleotide switch for genome editing. So this is the actual crystal structure, which is very nice. So it can be used from bacteria to humans. Sorry. So what are the applications? The applications are very broad. In agriculture, now people are genetically engineering plants so that, you know, they can grow under warmer temperatures. They can grow with less water, right? And more productive, longer shelf life, and so forth and so on. So a lot of industry and research behind CRISPR and plants. I'm still learning. Oh, could you click next? All right, cool. Oh, I skipped one. So one of the initial CRISPR experiments were done on dogs, making the skinny dog to be more buff. Micro pigs, these are pets. You make pigs that are smaller so that you can keep them as pets, $1,500, very cute. Hypoallergenic eggs, probably a lot more useful than micro pigs. And the anti-malaria mosquitoes, and we'll talk about the mosquito issue soon. Now, I want to highlight the Hercules, the buff dog here. I think you can realize that the potential of myostatin deletion causing increased muscle mass, you can help human cases, for example, with Duchenne's patients, where you have loss of muscle mass. That's a therapeutic use of CRISPR, right? But it's dual use. You can also theoretically make a Captain America, right? From Captain Rogers to Captain America. That would be enhancement. So this therapy and enhancement are dual uses of CRISPR, and we'll talk about this more later. Now, what are CRISPR gene drives? This is an interesting invention. So the idea is, if you remember your Punnett square from high school, which I don't, you have Mendelian inheritance. one allele from mom, one allele from dad. If you get one from dad, it's 50% chance. But with CRISPR, you can make it such that, well, the one allele you got that's edited, you'll double it to the other allele so that you become a homozygote, okay? So that way all your progenies will have it. That's one way to design it. So in a normal mosquito population, if you have a mutant, not many of them will be mutant, but with a gene drive, because it'll copy itself to the next allele, within a few generations, you'll have all that mutations in the mosquito population. So what you can do is that you can, because these mosquitoes, they carry diseases, right? Zika virus, malaria, dengue fever, you name it. Because of that, you can design mosquitoes such that if you mate with the mutant, they cannot have progenies. They might not be able to fly, or people do different modifications on the mosquitoes. And so you can drive species extinction using this method. Okay. The first U.S. open-air test of genetically modified mosquitoes deemed a success. This was the mosquitoes were released in the Keys in Florida. Though I saw in LA Times, they're also in California. So this deemed as a success. There are ways to control this so that they don't mutate. You can reverse the gene drive, you can kill them off, and so forth and so on. There are many technologies to control these CRISPR mosquitoes. Also, maybe they can drive away the New York rats. Poor New York pizza rat. Because this CRISPR-Cas9, this gene drive, have been shown to be able to be done in mice as well. So this is expandable in terms of what you can do, not just in lower or simpler organisms such as mosquitoes. So what are the issues surrounding gene drives and species extinction, right? So these are some ethical reflections, theological ethical reflections. Laudato Si reminds us that we are part of nature, not above nature. Who are we to do these things to other species, right? We need to consider the environmental effects of gene drives. This Lisa Fulham is also at GTU. All creatures, even microbes, have their intrinsic value. It's kind of an intrinsic value argument, independent of the relationship to us. Lynn White has brought this up, Chris McKay as well. Yet, there's a counter argument to this, right? That intrinsic values of other species are always relativized to humans when it comes to reality. In other words, okay, right? Like mosquitoes have their intrinsic values, but they're killing many people and causing diseases. So guess who wins? We win, right? So there is always, when the tire hits the road, this intrinsic value argument often goes out the window. We're back to instrumental ethics. Right? And the problem with instrumental ethics is that, well, circumstances, values change depending on the circumstances, right? You can have an E. coli that would be bad for your agriculture, but good for humans as a microbiome. Right? So you can't just drive it to extinction, for example. So it's very complicated. You don't have a strict guideline if you don't have intrinsic value. So I think Ted, for example, has published on responsibility ethics, trying to maybe get away from this dilemma and try to see, well, what is our responsibility in terms of a protectionist model to protect ourselves and all of creation. It's a different way of approaching the problem. And my approach is to say that, well, whenever we try to value these other organisms, other species, whether intrinsically or instrumentally, we need to look at their original context in their own microbiome and how they are related to the space that they occupy. Okay, this is coming more from my liberationism kind of post-colonial, right? Because organisms are usually tied to that land, tied to the seas in that specific environment. Science oftentimes harvest and colonize and mine these organisms for our own use. So there is an imperialistic aspect of science that we need to be mindful of. We cannot divorce them from their land. Oh, I lost my pointer. Okay. So are genetic models helpful? Yes, they are. I think even if not being applied to humans, genetic mouse models are very, very helpful. So this is an added volume, for example, of the use of transgenic mouse models for rare disease research. Because if you can copy the disease of a of a patient, the single mutation in the patient that causes the disease, you copy it in the mouse, then you can experiment on the mouse. Find the drug, right? Develop a new drug and then test the drug and see if it works on the mouse before you go back to considering using it in humans. So CRISPR is a very powerful research tool. And which one would we target first? And I published this in 2018. I would say, well, the monogenic diseases. There are about 4,000 monogenic diseases. Monogenic just means you can pinpoint the cause of the disease to a single gene, genetic mutation. That's all. Right? I mean, CRISPR technically can edit multiple genes at the same time because it's so easy to use. But when it comes to humans, let's just fix one at a time, right? But fixing one at a time, there are 4,000 of these diseases that are monogenic. So the opportunity is huge. This is about 200 million people globally that have these problems. Many of them are actually rare diseases. So rare diseases are not so rare. There are 7,000 rare diseases out there. And combined total, that's 300 million globally. So rare diseases are not so rare. The problem is it's difficult to get funding to do research on rare diseases because your impact score, your grant application will be not so great, right? Well, it doesn't help too many people. You know, developing this drug for companies as well will be very expensive. So how do we go about then to do these CRISPR gene therapy? In general, there are two ways. One is called ex vivo, outside of the body. You take some blood cells from the patient, you CRISPR them outside in lab, and you inject them back in. It's simple, ex vivo. You do the CRISPR outside. Or you can send the CRISPR into the body. Right? Then you would use viruses or nanoparticles to do the job. And you hope that, well, you of course design the virus to go to the specific target in the body to deliver the CRISPR-Cas system. Okay? That's in vivo gene therapy. So that's how you do it in one slide. This is a timeline in terms of history. I love these timelines because this is a way of scientists to reconstruct history. So it was identified CRISPR at 87. You study a bunch of structural functional relationships, and then in 2012 was the publication of CRISPR-Cas9, and you have the first clinical trial, CRISPR clinical trial against treatment of HIV 2017. And now we have a lot more, right? And 2020 was the Nobel Prize. If we look at now CRISPR clinical trials, you might think, well, do people really do this? Yes, they do. So over a dozen CRISPR clinical trials. You can go to clinicaltrials.gov. If you want to update yourself, you want to check, you're bored, you want to check how many CRISPR clinical trials. Maybe that's what I do for fun. And you can get a list. You can see the drugs, the mechanism, whether it's ex vivo or in vivo. We talked about that, right? And what clinical phase it is. Most of them are clinical phases 1 or 2. One of them is phase 3. Now, how broad is this gene therapy? Is this new? If you look at gene therapy in general, not CRISPR, CRISPR is just one of the newer tools that we can play with. But there are other genetic tools that are more difficult to use, but they were already made, right? So according to this paper, for example, as of 2017, there were already about 2,600 gene therapies globally. That's a lot. In 2021, 131 gene therapies, of which a dozen of them were CRISPR, right? By December 2022, a few months ago, about 27 cell and gene therapies are approved in the US by the FDA. But this is kind of the landscape in terms of what is going on. Of course, a lot of them are in the US and Europe. So we can cure disease by editing a person's DNA. Why aren't we? Why aren't we doing it more often? Right? This is an opinion article at New York Times by my previous professor, Fyodor Urnov, who's here at Berkeley. There are dangers, right? So let's talk about some of the dangers. Scientific risks. These are well known. Off-target effects, just like any drug. You send the guide RNA, it might bind elsewhere, not where you intended it to bind, right? So that's one possibility. 3 billion base pairs, 20, 30,000 genes. How do you know it'll bind in the right place? on-target effects. Just because you reach the destination, you delete that gene, it might have unintended consequences that you didn't know about before. So a classic example would be sickle cell anemia. We might get rid of sickle cell anemia, but then you will get rid of the resistance to malaria, which is a worse disease than sickle cell anemia, right? So we have to think of the on-target effects as well. Chimerism is probably more technical. If you send the CRISPR in, but not all of them get edited, then you end up with a chimera. And how well would this chimera be? You know, will it render the CRISPR modification useless, or will it be partially useful? Will it be overcome by the non-CRISPR cells, and so forth and so on? Epigenetic factors is, I think, very important. Epigenetic just means anything that's non-genetic. So if you think of our DNA, it has to be tightly wound to fit in the little nucleus, right? Because it's tightly wound, it's not always wound open, accessible. So you can put in a gene that'll make you smart. No, it doesn't exist, right? You can put a gene like that, but if the DNA is bound closed, it's not going to be able to be activated. It's not going to work. And so it's rendered useless. So just because you have the gene, you have to also consider the epigenetics. And nature has a way that usually epigenetically, if it doesn't want it, it'll repress it. I can talk more about that. It has tendencies like that. So there are strategies to overcome all the problems that I've talked about, right? So these the risks were from early guidelines from 2015 and so forth and so on. But people have been working on fixing these problems, right? So delivery issues, people were worried about viruses coming to our body. Well, what if the virus has gone wild? No, just kidding. What if our body reacts to the virus and have an immunogenic response? And this did happen in a gene therapy case in 1999, right, where a teenager passed away because of an immune reaction. So this is a serious concern. Now we can use nanoparticles. Nanoparticles are a lot safer, what's been discovered. I think people are shifting the nanoparticles. They're lipid balls, basically, that you deliver in. Okay. In terms of off-target, we can kind of anticipate what the off-targets using bioinformatics tools. There are also Cas9 nick cases. This is one of those things where you will need, usually CRISPR-Cas, they'll come in, they'll cut double-strand DNA, but you can make it such that they'll cut only one strand. That means you need 2 CRISPR-Cas to make the cut. In other words, if you're going to go wrong off-target, you have to have 2 CRISPR gone wrong, the same exact wrong side, which is highly unlikely, right? Because if you only have one of them gone wrong, it's just going to cut a single strand, nothing happens. DNA will fix itself, okay? So this makes it a lot safer. The downside is it'll be less efficient, because then that means you have to send in a lot more CRISPR-Cas for the targets that you actually want, because now you're going to need 2 CRISPR-Cas instead of 1. Right, so that's the drawback, but you can address the off-target effects. For the immune response, the other way to deal with it is you do the CRISPR modification in early in babies, so that you're tolerating, your immune system will tolerate that modification, so that you don't have an immune rejection. So there are many ways to overcome these problems, and I think we're getting more and more confident in experimenting with CRISPR and using CRISPR. All right, that's the science stuff. What about the non-science stuff, the application? Obama advisors urged action against CRISPR bioterror threat in 2016. When I first saw this, I thought, who's going to think of bioterrorism? You have to be pretty smart to know CRISPR, how it works. Well, I was wrong. I watched an episode of Designated Survivor. It was on Netflix already, where some biohacker made a CRISPR against people with dark skin. So you have the gene for dark skin, and the CRISPR will knock out certain genes and make it harmful for that person. So yeah, that was on Netflix. so much for ideas floating around. And this is actually idea put in practice. This is what Dr. Pickett mentioned. He-Jian Gui, Dr. He-Jian Gui, a biophysicist from China, he decided to do the human experiment modifications of a twin nicknamed Lulu and Nana to make them HIV resistant or AIDS resistant. Okay. So his thinking is, if I can design, right, CRISPR designer babies by definition is you design imply that if you do modification X, it'll cause a change in X. His idea is HIV needs this receptor, this hook on your cell CCR5 in order for it to attach. Well, if you get rid of the hook, the HIV can't attach. Voila, you're AIDS resistant. It doesn't work like that, of course. And the problem is CCR5 have their natural functions, right? So some studies in the mouse show that CCR5 might be good for to protect against genital herpes simplex. So you might get rid of HIV, but you might be more susceptible to HSV. Right? So again, this is kind of the on-target, unintended consequences that I talked about before. More papers were published. Well, maybe the CRISPR twins might have shortened lives because of the CCR5 knockout. And others will say, well, CCR5 suppresses cortical plasticity, which means flexibility in learning. So maybe the twins will be smarter because they don't have CCR5. So we don't know what'll happen, right? People speculate of what the possible unintended consequences are. And I think, you know, have we traded theological determinism for biological determinism, perhaps? We think, well, genes don't do everything, but if we know the environmental context, we can predict what'll happen. Right? That's kind of the spirit. If I just know your genomic makeup, what causes those 4,000 diseases, I CRISPR everything out to make sure it's perfect. And then you have a healthy diet, wear your Fitbit watch, walk 10,000 steps a day. Right? You should live up to 120 as long as you don't get hit by a car. That's some kind of a, you know, sociobiological determinism as well. Right? So are we truly free, or are we simply unconscious of our gene environmental predetermination? Regulations, how are these things regulated? Right. There are several bodies that have tried to write reports on this. American College of Medical Genetics and Genomics talked about the same scientific risks that I've discussed before, off-target effects and unknown epigenetic consequences. National Academies of Science, Engineering, and Medicine also did a report. They understood that there are advantages, right, prevention of inheritance of genetic diseases, advancements of basic science research. But again, you have the same four major concerns. Okay, and I think there was also a WHO report on this as well, more recent. But should someone decide to do this, who's going to stop them? That's the question, right? And this technique is easy to use. So gene editing tool went from labs to a middle school classroom. 8th graders learning how to do CRISPR and be able to do it. It's simple. That's why CRISPR is powerful. This biohacker, Josiah Zahner, he regrets it laterly, but he publicly injected himself with CRISPR. Right? So this is the term biohacker. So this is something that you can do in your garage poorly. You can do CRISPR very easy if you don't care about doing it properly and correctly. That's what I should say. It's easy in the sense that you just want to get it done dirty and quick. That's easy, but you don't, you know, but it's not clinically prudent. So CRISPR kit, $180 on Amazon, just to edit bacterial genome though. It arrives, I think, in 2-3 days, not prime yet. And all you need, I love this, you don't need anything else but water and a microwave. That's all you need. That's fun. So yes, this is easy to use. I've argued this. I think this is one of these technologies that are very powerful but easy to use. We're always concerned about access, right? But this is not like the race to the moon, where we need billions of dollars and a lot of technical expertise. This is something that's a lot easier for the public to access. With just some basic science and some protocol understanding, you can do CRISPR. So, America's first CRISPR law in California. Do not change your DNA at home. So that's the first law on that, other than the patent dispute. Sheila Jasanoff, who's contributing to our upcoming book, have promoted this idea of CRISPR democracy, gene editing and the need for inclusive deliberation. It's Benjamin Hurlbut, the son of Bill Hurlbut, who did a PhD at SDS at Harvard. That's how they're connected. Yeah, I think. Yeah. But yes, because this becomes a democracy, because everybody will be affected by this and should be mindful and should know about this technology. That's why I think a platform like this is very useful, where the public is also engaged. This is not just something that scientists can tell you what to do. This involves everybody. So this is a more elaborate diagram that I like from a recent paper. This is a way to think through on whether to use CRISPR or not in humans. Right? You can ask the question, does this disease that you want to fix have a known and monogenic cause? Does it have a single gene as a cause? If no, do something else. If yes, well, is this disease bad enough? Is the mortality high? Is the morbidity high? Right? If yes, then you consider the ethical and legal considerations. Is there a societal consensus on this? Have measures been taken to reasonably reduce the risks? Is there consent? This is also very important, the idea of autonomy, right, in the beneficence diagram, autonomy, beneficence. Is it suitable? Is there a suitable long-term monitoring of the patient? And are there any conflicts of interest? This is the legal aspect, ethical aspect. The scientific aspect, have this been done in animals? Are there other treatments? If yes, are the other treatments better than CRISPR or the same? If yes, then why are we doing this, right? If there is, and it's not as good, well, is CRISPR a lot more expensive? Is that treatment more expensive? Cost-benefit analysis, right? So these are the kind of the scientific input, technical input. And if you say yes to all these things, and maybe we can consider CRISPR as a therapy, but still not enhancement. Right? All right, so several themes to consider. Natural versus modified, therapy versus enhancement, somatic versus germline, okay? So we'll talk about the first one first. Are we playing God? Of course, I think this is one of Ted's book title. Are we playing God? Should we alter nature? Is this a Promethean, Frankensteinian hubris? Like according to Leon Kass, who picked up on Hans Jonas on this issue. There's a reason why nature is the way it is. Don't tinker with it, says Rifkin. Right? So these people would say, no, do not do this. We're playing God. Do not alter nature. But there's a counter argument. Well, you're assuming that the way nature is the way it should be. Right? This is a Hume's guillotine, the is-ought distinction. Okay, maybe with G.E. Moore, it becomes a naturalistic fallacy. Right? Is nature how things should be? Or as being refined by Lepaxis, had told me, does nature define what is good? That's also a form of naturalistic fallacy. Maybe we're not playing God, Phil Hefner says. Maybe we're playing with God as created co-creators. Okay? We're given this tool. We might as well be active and consider how this tool can be used for the greater good. There's also the idea that nature is incomplete, right? There's an openness, maybe from Terry Deacon here, but also from Ted, that the future is open. So we can participate in designing that. And I think there is a blurry distinction even in science, I would say. You know, we often think, well, you're doing this CRISPR modification. This is unnatural. Fixing mutations is unnatural if you put CRISPR in. But nature fixes mutations on its own. It's called revertant mutations. Right? So this is a disease. Originally, it's GC. Your first mutation, you have a disease. But sometime, you have a second mutation that put you back into the first one, and you get better. Okay? So isn't CRISPR just helping Mother Nature to fix herself? Okay? Is the question. So this natural versus artificial, I think, is a blurry line. And what is nature, anyways? This is more a question for you, not for me, right? Are we our genes? Should we alter our DNA? No. What about identical twins? They have the same DNA, but obviously different persons. Well, they're different persons. Is it because of their brain? So maybe the personality is in the brain. You tinker the genes of the neurons, then you're tinkering with human nature. Or are we nodes within social infrastructure? The brain is controlled by interactions with the environment. So maybe it's okay to tinker everything that's in our body. It's a society that kind of defines who we are. Maybe this is more of a Marxist, analysis. Or to be theological, to be Parthian. Are we how we respond to God? So there are different ways to look at this question, and I think we have to be holistic in assessing this question. Therapy versus enhancement. I've been saying this for a while because this is an issue that bugs me a little bit. What is normal? If we think of what is normal, the normal is usually kind of like the mean. Right? If you're below the mean, you're sick. Right? If you can't walk, for example, you're sick. If you can run very fast, well, you're great. You're very healthy. You're enhanced. Right? So a treatment to push a person's capability from below the mean to mean is considered therapy. A modification to push a person from the mean to above the mean would be considered enhancement, right? But the problem is, of course, if you provide therapy is allowed, enhancement is not allowed. Well, if you provide therapy, the mean's going to move. If the average human height is 5'10", you do CRISPR, then for people who are shorter than 5'10", then of course the mean shifts to 6 feet. I mean, you can't do this, but that's the general idea. So this therapy enhancement distinction is either non-existent, as some philosophers have argued, or at least a moving line. That would kind of my position. I think there is a line. We do have some sense where certain things are enhancement. I would even call them cosmetic. Okay? There are certain things that are also therapy. That's very obvious. But the line does get blurry. And not only thinking of a line, but we need to think of what kind of population and context are we talking about. So, for example, if there's a genetic modifications on me to make myself radiation resistant, that's an enhancement. I'm resistant to nuclear bombs. But for an astronaut who needs to fly to outer space, being exposed to radiation, that's not enhancement. That's a preventative therapy. They need that for their job. That's an essential genetic modification. Right? So this idea is also from Conrad and Braden, who are writing in our book. So the context also alters what we mean by therapy and enhancement. It cannot be universalized to everything. We need to look at specific context as well. All right, somatic versus germline. Germline basically just means you can do the CRISPR modification either in your sperm or your egg. If you do that, then basically every cell in your body will have that genetic modification, which also means you'll pass it out to the future generations, right? This is one of those things that change your whole descendants, your generation. Somatic mutation is if you do CRISPR on me now, just not on my gametes, then it'll stay with me. The CRISPR on my liver, then it'll stay in my liver. That's it. Okay. So this is the distinction. And right now, the guideline, well, we can talk about the guidelines. In general, germline, you have to be more cautious. Somatic, that's fine. Okay. For obvious reasons. Maybe not so obvious, according to Eric Youngst. Right? Even germline therapy, there are cons, there are risks. It's a slippery slope to enhancements, right? We don't have the consent of future generations. We're tinkering with their genome. Allocation of resources, maybe we can do other things. And integrity of genetic patrimony, right? The future generations have the right to inherit the original genome, not the one that we modified and want to give to our kids. But there are pro arguments as well. Utility. The germline gene therapy, maybe, it's offering a true cure for diseases. Maybe it's essential. Maybe that's the only cure. The only way to fix it is if you do a germline. Oh, did I click that? Thank you. Maybe prevention in terms of germline therapy is less costly than a lifetime of treatment. Right? This would be a pro argument for germline. We talk about the autonomy and the consent of the future generation, but what about the parental autonomy? What about the scientific freedom? So these are kind of pros and cons that have been posed by Eric Youngst. All right, so if we survey, this is a very important paper by a Canadian philosopher thinker, François Baylis, published in CRISPR Journal 2020. He surveys many countries, 96 countries, policies on human germline genome editing, not for reproduction, and you get 19 countries prohibiting, 11 permitting. US is green, UN permits germline genome editing, but not for reproduction. Right? Now, what about for reproduction? No, everybody says no. And then you have 70 countries prohibiting. 5 countries are making exceptions. That would be Belgium, Colombia, Italy, Panama, and United Arab Emirates. That's interesting. All right, so should we enhance our children? I guess the question, right? Will we be blamed if we edited their genome because they want their original genome? Will we be blamed if we did not edit their genomes? When you do it, Dad, and I'm shorter than everybody else. Okay. I think as parents, we're going to be blamed either way by our children. If the technology is available, we are responsible to decide, right? To not act is to have decided. On the other hand, there is this past history of the possibility of eugenics that we have to be mindful about. And then John Slattery writes a book chapter on that in our upcoming book. And there's the issue of commodification of children. When we're editing our kids, is it really for them or is it for us? Well, our kids are smarter, our kids are healthier, and so forth and so on, right? Then we probably have commodified our babies. If gene editing goes wrong, do we get a discount on the kid? Do we then make another kid, the perfect child syndrome? Well, modified X, he's brilliant, but not good in sports. Let's modify X and Y, have a second kid, have a third kid, right? So forth and so on. And I would say, at what point is the enhanced child no longer my child? My son looks like me, but if I had edited him to be, 6'5", blonde with blue eyes, how's that my kid, right? So the parental aspect of it is still important, as Young's pointed out. All right, some theological reflections, it usually comes in within the framework of dignity, humana vitae. So in some cases, for example, from Austriaco, he's a Dominican priest and also a scientist. He did his PhD at MIT. He said something like, maybe it is okay to do germline modification if it is done safely. It's preventative therapy, not enhancement, and it still does not undermine their dignity. That could be acceptable. Right? So people are more open to this idea. Lisa points out in terms of the dignity that we can't think only of the dignity of the embryo that we're CRISPRing, but someone has to carry out the pregnancy, and these are women. There's more of a feminist perspective. We need to think about what is the impact of such CRISPR therapy in the mom. Okay. So dignity can go in many different directions. All right, concluding remarks. CRISPR applications are very broad. These are the scientific dangers, off-target, on-target effects, and epigenetic factors. The CRISPR regulations are there, but they're not enforceable. And they are somewhat unclear on where to draw the line or on what the line means. And by line, I meant natural versus modified and theology versus enhancement. Right? With the somatic versus germline, that's pretty clear. And I think CRISPR democratization, because it's so easy to use for everybody, it really calls for a public engagement at a platform such as this, right? Where CRISPR applications and regulations can be discussed with the public. Thank you.
Well, thank you, Arvin, very much for that. What I would like to do here in a couple of minutes of response is raise the question, why is knowing something about this kind of science, important for the pastor, the pastors of today and the pastors for tomorrow. I just have to say that I've really appreciated hearing Arvin here today. It was some years ago when Arvin was studying genetics at Cal across the street that he showed up in a doctoral seminar on systematic theology, and he's been on 2 tracks ever since. He didn't mention, or Ray didn't mention either, that he's a cancer researcher, and up until recently he had 2 laboratories going, one at Stanford and one at Cal. And I'm hoping that there will be a day in which he'll receive the Nobel Prize for curing cancer. You know, you got to hope, right? Why might CRISPR gene editing be important for a pastor to know about? Well, there are 2 areas. The first one is pastoral care. Over recent decades now, families who are expecting children will frequently go to a genetic counselor. The OBGYN will recommend one. And then they show up in the pastor's office with what they have learned. Amniocentesis tells them something about the genome of the child that's about to be born. And for a long time, the question was, should we abort or not? Why? Well, they found one of those monogenetic genes that are responsible for disease. And do you want to get rid of the child before the child's born because they're going to suffer? Certainly in Jewish families with Tay-Sachs, lots of rabbis find themselves counseling people in that circumstance. There was a graduate of PLTS some years ago named Martha, and she became a pastor up in Oregon, and she and her husband decided to go with in vitro fertilization, and their baby was born, and then upon birth, the child had they discovered the child had Williams syndrome due to a malfunctioning allele on chromosome 7. Well, the symptoms were that he'd walk down the hall and he'd bump into the walls, and he would have a shortened lifespan and a few things like that. So here they are using exotic reproductive technologies. Had they known that the child had a gene for Williams syndrome, would they have aborted? Or if it were to happen now, might they have been able to alter chromosome 7 using CRISPR gene technology? And if so, Pastor, what should I do? Well, it's good if the pastor has a little knowledge of what's going on here in those kinds of circumstances. One of the issues that's almost always on our minds, as Arvin had mentioned, is this relationship between nature and altering nature. The romanticism in us tends to say, if it's natural, it's good. And when we get into the human genome with wrenches and screwdrivers and try to change it, are we playing God? Are we violating nature? Are we risking Frankenstein or Prometheus or something of that particular nature? Those questions hover around our consciousness. And, you know, sometimes a pastor's a good person to ask about this kind of thing. And so having a little bit of knowledge and actually having already formulated some opinions is a good idea for pastoral care. The other item near the end of Arvin's presentation has to do with public policy. When in China, this researcher, as Arvin mentioned, decided to go ahead and bring into the world 2 babies whose genome had been altered by CRISPR because he wanted to prevent them from contracting the AIDS virus. And he was very proud that he had achieved this technology. And the whole world of ethicists and genetic researchers rose up in anger at this. Why? Well, the first thing is that those who oppose this bringing children into the world with an altered genome, the first problem is that genes work in systems. Arvin used the word context. One gene can make 4 different proteins, depending on its interaction with the other genes. And if you just alter a gene like that and you don't know the context, you don't know how it works systematically with other DNA, you could throw things out of whack. And that's what happened in this case. The critics were able to show that the lifespan of these children is going to be shortened now because of the, even though the work was on target, the consequences are such. And so my experience with geneticists is they're saying, we don't know enough yet. And so let's not experiment on a human being. A human being has dignity until we've got all of the factors in hand, and the geneticists don't at this particular point. And so you heard Arvin saying towards the end there that if you and I engage in somatic therapy, that is to say, if you've got a liver and that's a problem, and CRISPR gene therapy can lead to healing, if not regeneration, good, go right ahead. But that will, even though that affects you therapeutically, it's not going to affect the children you bring into the world. That's where, for the most part, ethicists and geneticists have the big concern. So the traffic is going to be slow on this technology, even though the technology is easy. But as Arvin was saying, the easiness of it may turn out to be a difficulty, because any teenager in a garage could start altering the genomes of the dogs and cats in the neighborhood. And we don't know if chaos is going to be the result of this. So if you're a pastor, check the neighborhood and see if the dogs and cats are starting to look different because of this. It's also the case that in many communities, pastors are asked for their opinions on policy matters. And so having some level of keeping up to date with the science is always good for just that level of sophistication that pastors have to offer the community. Now, as an aside, I was working yesterday on a movement that you probably don't know about called Don't Trust Your Pastor. And in noticing that, the Gallup poll reports that trust in pastors has dropped to 36%. Who's lower than pastors? Telemarketers are lower. Yeah. Who's higher? Nurses, doctors, and pharmacists. What's wrong? Why is it that the public does not trust the ethical profile of the clergy? I would just say that in some of these areas, such as we've been talking about today, upping the scientific knowledge and the ethical discernment on subjects like this might be a way of actually improving the professional contribution our average pastor can make to the wider community. Well, those are my thoughts, and I want to thank Ray and PLTS and the Mohrenweiser family, I don't know if I pronounced that correctly or not, for their farsightedness, because this interaction between faith and science should not be dominant, but it is a way of enriching the ministry of the pastors and leaders of our church. So thank you very much.
Thank you, Ted. First of all, I want to thank you, Arvin, for, an incredibly clear presentation and a very balanced presentation where you look at the pros and cons and they're almost equal on each one. So you've definitely raised more questions than we can answer. And I have a lot of questions, and I'm going to invite people to ask questions and you to come up. But I think about the thing I think most about from this is just, our inclination to want to control things, and we all know this from our own experience, the unintended consequences of trying to control our lives or anybody else's lives. So you've given us a lot to think about, and I appreciate, as Ted said, you sort of educating us as theologians and pastors, because this stuff is out there, and I'm not sure how much people are aware of it, but it raises all kinds of questions about even what it means to be human, as you say. So thank you so much. Yeah.